Alright, so in today's tutorial video, we want to look at statistical methods for analyzing one sample data in Python using Jupyter Notebook. And if you have not watched my previous tutorial video on the flowchart for classifying data, I'll urge you to kindly take a look at that before you jump into this, because in that video tutorial I have illustrated and explained the data structure for one sample data, which will help you to catch up with everything that I'm going to illustrate in today's video tutorial. Okay, so let's get started. Basically, anytime you have one sample data, there are two statistical methods or techniques you can actually use to analyze such a data. We have the parametric test and we have the non-parametric test. Okay, so the parametric tests are basically statistical tests for population parameters such as the mean, based on the assumption that the population from which the samples are selected follow a normal distribution. Okay, and the non-parametric tests, on the other hand, are statistical tests for population parameters such as the median, that is not based on the assumption that the population from which the samples are selected will follow a normal distribution. Okay, so the type of parametric test that we often use in analyzing one sample data is called a t-test or the one sample t-test, and the non-parametric test we often use in analyzing the one sample data is called a sign test or the quantile test. So let's take a look at the parametric test for one sample data, which is the one sample t-test. So the one sample t-test is a statistical hypothesis test used to determine whether a known population mean is different from a specific value. For example, we might want to know if the mean waiting time in a doctor's office is greater than 1 hour, or if the mean growth of roses is 4 inches or more after 2 weeks of treatment with a certain fertilizer. Now, before we make use of this test, we have to basically satisfy some assumptions. One of the assumptions is that the data must be continuous, and also the data must be normally distributed. Okay, now let's take a look at the non-parametric test for one sample data, which is the sign or the quantile test. So the sign test is basically a statistical hypothesis test used to determine whether a known population median is different from a specific value. For example, we might want to know if the median waiting time in a doctor's office is greater than 1 hour, or if the median growth of roses is 4 inches or more after 2 weeks of treatment with a certain fertilizer. There are some conditions that must be satisfied before we make use of this test. One of the conditions is that the data must be continuous, and also the data must not be normally distributed. Okay, so now let's take an example. So using the iris dataset, as a researcher, you wish to test whether the average length of the sepal from the iris dataset is different from 6 centimeters. We want to perform a statistical analysis for this data. Okay, so let's take a look at the iris dataset. So this is the iris dataset in the CSV format. Okay, so this is a clean data. Okay, let's assume we have already cleaned or we have prepared our data for analysis. Okay, anytime you have a data set, you have to make sure that you clean or you prepare it so that it can be used for model building or for your analysis. Okay, so I'm going to leave this data file in the description of the video so that you can download it. Okay, so this is the iris data set, and I want to find if the average value of the sepal length is different from 6. Now, if you just use or make use of this whole variable, right, this in your analysis, maybe you want to use this sepal length values to determine whether the average is different from 6. Okay, if you just make use of this in performing your t-test or your quantile test, you may end up doing a wrong thing, because you have to take note that the sepal length was taken for 3 different groups of the species. Okay, so first of all, we have to filter the sepal length for setosa. You also have to filter the sepal length for versicolor, and we also have to do the same for virginica, and then perform a t-test or a quantile test on each one of them. Okay, so that's basically what we're going to do in our data manipulation. So let's jump into our Jupyter Notebook and let's get started. So statistical analysis for one sample data in Python, you're going to make use of the Jupyter Notebook. So first of all, we want to load the library that we're going to use for analysis, which is the pandas library. If you don't know how to make use of this library, you can go watch my previous tutorial video on exploratory data analysis in Python. Okay, so that you can know how to install this and actually get some ideas with regards to some of the libraries. Okay, so we're going to load this library. So I'm going to run this. Okay, So once it's done, you'll see a number in this box, so it's still loading. Now once we are done importing this pandas library, we can then load our data set. So I'm assigning my data to idat. Okay, so from pandas library, I'm using the function read_csv, which will help me to import a CSV file. Okay, so the name of the data is clean data, and make sure that this data file is in the same folder where you have your Python source file. Okay, so let me show you. So this is my Python source file, right, in the same folder where I have my data file. Make sure that they are in the same folder. Okay, alright, so let's go back. So since this data file is in the same folder where my Python source file or the script file is, I don't need to set a working directory, so I can just run this. Alright, so we can view our data set. So let's run this to view the data set. Alright, so here we go. So we can see in here we have the first 5 observations and the last 5 observations. Okay, so there's a separ length. Now let's get more information with regards to the data set. So let's get a structure of our data set. So let's run this. Alright, so here we go. We have 148 entries indexed from 0 to 147, and we also have 5 columns indexed from 0 to 4. So you can see that the separ length, separ width, petal length, and petal width, they are all float, that's the data type, which are basically numeric. And we can also see that that of the species is an object. Okay, so in our data manipulation, we are supposed to convert this into a categorical variable, or basically we are supposed to assign values to the labels, right, of this variable. So let's get that. So in our data manipulation, we are basically going to recode a species from string to numeric, or basically assign some values to the labels. Okay, so for my imported data idat, I'm picking the variable species, and I basically want to convert that variable into a category, right, or into a categorical type. Okay, so let's run this. Alright, so now let's look at the structure of our data set now. Alright, so we can see that now the species has been converted into a categorical type. Okay, so we are good to go now. One thing that we can also do is to basically rename the variables if we wish to. So let's see how we can do this. Let's say I want to rename the variables. So I'm assigning my data now, my data set, to a new dat. And so for my imported data idat, I want to rename the columns. Separ length as SL. I want to basically rename separ width as SW, petal length as PL, and petal width as PW, and also species as SP. Okay, so let's run this. Alright, so now let's look at the structure of our new data set. Alright, so you can see that now we have renamed the variables. Okay, so we are good to go now. Alright, so now that we are done with these steps, we can then proceed to filter our data. Remember that we are asked to find, or want to find, if the average value of the separ length is different from source. But we know that the separ length was taken for 3 different groups of the species, so we have to filter those out. Okay, so let's get started. So I want to filter the setosa of the separ length, right? So I'm assigning my data now to G1 underscore SI. That's the group 1 of the separ length, which is basically setosa. Okay, so this is basically what we're going to do. We are going to pick from this code of line that we have. We are basically going to filter the separ length of the setosa. So basically we are going to pick these values from 5.1 to let's say, let me scroll down to 5. So it's going to pick these values. Okay, that's for setosa. Okay, then we will do same for vesicular. We also pick that values from 51, observation 51 to let's see, to that of 100. Okay, it ends here. That's for vesicular. Then we will do same for virginica. Okay, so let's go back to our Jupyter Notebook. So for my new dat, I want to pick the variable species, and I'm interested in the label Setosa. I want to filter the sepal length, okay? Then I want to basically see the first 5 observations, so I'm using the dot head, okay? Let's run this to see the result. Alright, so here we go. So this just give me the first 5 observations. Now we can do the same for versicolor. I want to filter the versicolor of the sepal length, so let's get this also done. So I'm assigning my data now to gene2_sl. So for my new that I'm interested in the variable species, and I'm picking the label versicolor, and I'm filtering the sepal length variable, okay? So let's run this. Alright, so here we go. Then let's do that for Virginica. So now I'm assigning my data to gene3_sa. That's the group 3 of the sepal length, which is Virginica. So for my new data, I'm picking the variable species, and I'm interested in the label Virginica. Then I'll filter the variable of sepal length, okay? So let's run this. Okay, so here we go. Now that we are done with this, we can proceed and do some exploratory data analysis, okay? Please, if you have not watched my previous tutorial on UDA in Python, I urge you to kindly watch that if you want to get more insight or more information with regards to UDA, okay? So in our exploratory data analysis, we want to start with some summary statistics of the sepal length for Setosa, for versicolor, and for Virginica, okay? So let's get that of the summary statistics of the sepal length for Setosa. So let me run this. So in here I get some information with regards to the count. That's the number of data points in this data, right? So we have 49. We also have the mean value to be around 5 centimeters. This is basically where most of the data points are being clustered, okay? That's the peak of our data. It's around 5 centimeters. We also have the information with regards to the variability in our data set, which is given by the standard deviation. Okay. Then we also have information with regards to the 5-number summary. The first item in the 5-number summary is given by the minimum value, and the second item is given by the 25th percentile or the first quartile, and the third item in our 5-number summary is given by the 50th percentile, right, or the median value. Then we also have the fourth item in our 5-number summary to be given by the 75th percentile or the third quartile. We also have the last item as the maximum value, okay? So the minimum and the maximum value will help us to get a range. Once we subtract this from that, subtract the minimum value from the maximum value, we get a range. And the range basically tells us the overall dispersion of values in our data set, okay? Then with the 50th percentile, basically is the same as the median. So once we divide our data into 2, right, we know that 50% of the values are below 5 centimeters and 50% of the values are above 5 centimeters, okay? And the median of the upper half is given by the 75th percentile or the third quartile. So we know that 25% of the values are above 5.2 centimeters and 75% of the values or the data points are below 5.2 centimeters. So we can interpret this as the top 25% of the sepal from the Setosa plant increases in length than 5.2 centimeters, okay? And also the median of the lower half is given by the 25th percentile or the first quartile. So we know that 25% of the values fall below 4.8 centimeters and 75% of the data points are above 4.8 centimeters. So we can also interpret this as a single Setosa plant or the sepal chosen at random has a 75% chance of increasing in length than 4.8 centimeters, okay? So before you start with your main analysis, which is the t-test or the quantile test, you have to basically give some descriptive statistics, okay? You have to basically get some summary statistics of your data, okay? So these are, maybe you can interpret some of your numerical summaries before you start with your main analysis. Now let's get the summary statistics for the versicolor data. So let's run this. Okay, so here we have the number of count to be 50. We also have the mean value, standard deviation information with regards to the 5-number summary. So we can give the same interpretation to this. Let's get the summary statistics for the Virginica data. Alright, so here we go. So We also have the same information here. Alright, so another thing that we can also do in our exploratory data analysis is to basically visualize our 5-number summary making use of the box plot. Okay, so I want to visualize the 5-number summary using Boxplot. Okay, so the Boxplot is basically a way of displaying the distribution of the data based on the 5-number summary. Apart from getting information with regards to the 5-number summary, we also get to know how the shape of our distribution will look like. Okay, so before we visualize our 5-number summary, let's load some libraries for data visualization. We need a matplotlib. Okay, so if you don't have this installed, you have to install it. That is why I'm urging you to watch my previous tutorial video on GDA so that you can get to know how to install these libraries. Okay, so you have to first of all install this if you don't have it installed by making use of the pip function. So we have already installed this, so I'm just going straight to use it. So from the matplotlib, I want to import the pyplot as plt. Then I also need some library, that's a Seaborn library. So I want to import Seaborn library as SNS. Okay, so let's run this. Alright, so I want to get a box plot or a sepal length for Setosa. I'm assigning my graphic data to P1. Right, that's my box plot to P1. So for my Seaborn library, I'm making use of the box plot function. The variable of interest is SL, that is sepal length, and my data is J1 underscore SL, that's the data for Setosa. So let's run this. Okay, so it's still loading. Alright, so here we go. So this is the box plot. So we can see the first item of the 5-number summary being given by this point, right, which is pointing towards 4.4 centimeters. The second item of 5-number summary is given in this item, right, pointing towards 4.8. The median value of the 50th percentile is given to by this line, the median line. Then we also have the third quartile shown at this point, right, and the maximum value is shown at this point, right. This is the item pointing toward the maximum value. So aside getting information with regards to the 5-number summary, we also get to know the shape of our distribution. If you look at the distance from here, the minimum value to the first quartile, and if you take the distance from the third quartile to the maximum value, you can see that they are almost the same. The distance is not all that much. If you look at the shape or the area of this, the area in this box that's on the right hand side compared to the left hand side, you can see that they're almost the same, right? So this is an indication that the data of the sepal length for Setosa is nearly symmetric. Okay, we know that a distribution is said to be symmetric if it has the same shape on either side of the center value. So if you look at this distance from here to here, and if you also compare the area in this box compared to the area on the right hand side, right, you can see that they're almost the same. So we can say that this data is nearly symmetric. Let's check for that of the vesicular. So the box plot of the sepal length for vesicular, I'm assigning my data, my graphic data to P2. So from the Seaborn, I'm making sort of function box plot. The variable of interest is sepal length, and the data is G2 underscore XL. So let's run this. So in here too, we can see that the shape of the distribution is nearly symmetric, right? So you can see that the distance from the minimum value to the first quartile is not significantly it's not different from the distance from the third quartile to the maximum value. And you can also see that the area in this box on the right hand side is slightly bigger than the area on the left hand side in this box. So looking at this, we can also say that the data of the sepal length for vesicular is nearly symmetric. Okay, let's check for that of the virginica. So the box plot of the sepal length for virginica, let's run this. So let's see, here we can see some extreme values. Right, having extreme values does not necessarily mean that they are outliers. You have to be very careful when you're dealing with outliers. Okay, so in here too we can see that the shape of the distribution is nearly symmetric, right? Even though the area in this on the right hand side of the box is larger than the area on the left hand side, but you can see that the distance from the minimum to the first quartile and also the distance from the third quartile to the maximum value, they are almost the same, right? So we can also say that this is nearly symmetric. So in order to be sure with regards to the shape of the distribution, because we need this so that we can know whether we are supposed to make use of the t-test or the quantile test. So this exploratory data analysis is very important, giving us some information with regards to whether our distribution or the data that we have at hand is symmetric or not symmetric. So in order to be sure with regards to the shape of the distribution, we can even compute for the skewness and kurtosis. We know that skewness basically measure the degree and direction of departure from symmetry, right? And for kurtosis, we are basically looking at the tails or the thickness in a curve, right? That is how heavy or light the tails of the distribution look like. So we can do that in Python. We want to compute for skewness and kurtosis, but we need a library. Right, so we are going to make use of the scientific Python library, in short as the SciPy library, okay, or the CPy or the SciPy library, okay. So we need this library to help us compute for skewness and kurtosis. So we want to import that. So from the SciPy start library, we want to import the skew function, and from the SciPy start function, we want to import the kurtosis, right? So let's run this. Alright, so once we are done with the importation, we can now compute the skewness or the sepal length for setosa, and we can also compute that for vesicular, and we can also get that for virginica. So let's run this. Okay, so here we go. We can see that all the skewness is within the range of minus 0.5 and plus 0.5. Now, according to literature, if the skewness coefficient is within this range of minus 0.5 or plus 0.5, then it means that the data is nearly symmetric, okay? But if the skewness coefficient is greater than plus 1 or less than minus 1, then we can say it's highly skewed. So from this result, we can confirm that the data that we have at hand, right, that the data of the sepal length for setosa, for vesicular, and for virginica, they are nearly symmetric. So this is basically telling us that if we make use of the t-test, we are going to end up making a correct inference or the right inference, okay? So now let's look for or check that of the kurtosis. In here, we're making use of the SS kurtosis, okay? The SS kurtosis is basically used in comparing the kurtosis coefficient with that of the normal distribution. We normally subtract 3 from the kurtosis coefficient, but in Python, the result that is going to be displayed is actually the SS kurtosis, so we don't have to subtract 3 from the kurtosis coefficient, okay? So this first code of line is going to give us the SS kurtosis of the sepal length for setosa, right? And this is going to give us for that of vesicular, and the last one is going to give us the SS kurtosis of the sepal length for virginica. So let's run this. Now we can see that the SS kurtosis, they are all less than 0, which basically means that the distribution is platykurtic, right? Which basically means that the thickness in the curve is flat and spread out, or the tails of the distribution is flat and spread out, okay? If the SS kurtosis were to be greater than 0, then we will have a leptokurtic distribution, which means that the tails or the thickness in the curve is basically high and thin, or the tails of the distribution is high and thin, okay? Now if the SS kurtosis were approximately 0, then we can say it is mesokurtic distribution, which basically means that the thickness in the curve is normal in shape, okay? So we can also get more insight with regards to the skewness and kurtosis by making use of the histogram plot. So let's check that one out. So histogram plot of the sepal length for setosa. I'm basically assigning my histogram plot to H1, that is for setosa. So for my pie plot, I'm making use of the histogram function. I'm interested in a variable of for my setosa date, I'm interested in a variable of sepal length, and I'm setting the bins to 10. The bin basically is a single bar which represents a range of values, and the height of the bar represents how many data points fall in that range. So you can change this if you wish to. The color for my histogram, that's the bins, is going to be, I want to fill the bins with a cyan color. Then the edge of the bars should be green. Okay. Then I also want to add the mean value right in my histogram plot so that I can use this to check for the skewness, right? And I want to give the color for the mean value as red, and the line style should be dashed. Then I also want to use the displot function from the Seaborn library to check for excess ketoses or ketoses. Okay. So in here the variable is sepal length, and the kernel density estimate is going to be true so that I can see the density curve within my histogram plot. Then the bins should be 10. My data is J1 underscore XL, which is basically the data for setosa. Okay, so let's run this to see the result. Alright, so if this is my middle value or the mean value, right, then if you look at the weight of the distribution on the left hand side compared to the weight of the distribution on the right hand side, they are almost the same. So we can say that this is nearly symmetric. Now in terms of ketoses, I can make use of the density curve within my histogram plot. So if you look at the density curve, you can see that the tails of the distribution seems to be flat and spread out. So that is why this is said to be a plasticated distribution. Okay, let's check that off the histogram plot of the sepal length for vesicular. So now I'm assigning my histogram plot to H2. So for my entire plot, I'm making use of the histogram function. So my data, I'm interested in the variable sepal length, right? The bins is 10, the color now is green, edge color should be yellow. Then I also want to plot the mean value within my histogram plot to check for skewness, right? And my color is red, the line style is dashed. Then I also want to make use of the displot function from the Seaborn library to check for excess ketoses. Okay, so let's run this to see the result. Alright, so in here, if I'm to divide my data into 2, right, this is the mean value, you can see that the weight of the distribution on the left hand side is the same as the weight of the distribution on the right hand side. So we can also say that this is nearly symmetric. And in terms of ketoses, right, as ketoses, we can see that the tails of the distribution seems to be flat and spread out. That is why this is said to be plasticated distribution. Okay, let's check for that of the histogram plot of the sepal length for Virginica. So now my histogram plot, I'm assigning this to H3, right? The data is that of Virginica. I'm interested in the variable sepal length. Now the color for my bins is going to be blue. The edge of the color is going to be black, right? And I also want to plot the mean value within my histogram to check for skewness, right? And I also want to use the displot function to check for excess ketoses. Okay, so this is going to show the density curve within my histogram plot to check for that of the excess ketoses. So let's run this to see the result. Alright, so once I scroll down, if I'm to divide my data into 2, this is the mean value, right? And the weight of the distribution on the left hand side is almost the same as the weight of the distribution on the right hand side. So we can say this is also nearly symmetric. And now let's check for that of the excess ketoses. From the left hand side, we can see that it was approximately normal, but heading to the right hand side, you can see the density curve seems to be flat and spread out. That is why this is also said to be plasticated distribution. Okay, now that we are done with this exploratory data analysis, we can now test for the normality assumption, because now with the information that we have obtained from the EDA, we can basically confirm that the data that we have for setosa, for vesicular, and for Virginica, right, that is sepal length data, they are almost symmetric. So we can basically test for this normality assumption. We already know that the data is continuous, right? If you check this, you can see that they are continuous. Okay, that's the sepal length, they are continuous. But one assumption that we also have to check is the normality assumption. Okay, so let's do that. And with the normality assumption, we have three most frequently used tests that we want to make use of. We want to make use of the QQ plot, the Shapiro-Wilk test, and the Anderson-Darling test. Okay, so let's start with the QQ plot. The QQ plot basically also known as the quantile plot, draws the correlation between a given sample and the normal distribution. Okay, such that if the data is approximately normal, the plot will closely resemble a straight line sloping upwards to the right. But before we make use of the QQ plot, we need a library. Okay, from the SciPy library, we want to import the stats function as st. Then we also need a PyLab library, which basically provides some interface to the matplotlib. Okay, let's run this. Okay, once we are done with importation, let's get a QQ plot or a sepal length for Setosa. So I'm assigning my plot to Q1. So from the start function, I'm making use of the probability plot, and my data is that of Setosa. I'm interested in the variable sepal length. The distribution is normal, right, or norm. Then the plot should be PyLab. Okay, let's run this to see the result. Alright, so if we look at this plot, we can see that the plot closely resembles a straight line, right, sloping upward to the right. So this is an indication that the data of the sepal length for Setosa is normally distributed, and therefore making use of the t-test is required. Okay, let's check that of the QQ plot of the sepal length for versicolor. So let's run this. So now I'm assigning my plot to Q2. Right, the data is now that of the versicolor, and the variable is sepal length. So let's run this. Okay, so if we look at this plot, we can also see that it closely resembles a straight line sloping upward to the right. So this is an indication that the data of the sepal length for versicolor is normally distributed, and therefore making use of the t-test is required. Let's check that of the QQ plot of the sepal length for Virginica. Okay, so now my plot, I'm assigning it to Q3. Right, the data is now that of Virginica, and I'm picking the variable of sepal length. So let's run this. Alright, so we can see that even though just some few points seems to deviate from the straight line, you can say that most of the points, right, or the plot basically resemble a straight line sloping upward to the right. So this is also symmetric, and therefore making use of the t-test is required. So another test that we can also use to confirm this graphics, right, is to make use of the Shapiro-Wilk test or the Anderson-Darling test. These are other normality tests that we often make use of. Okay, so the null hypothesis for the Shapiro-Wilk test and the Anderson-Darling test is that the data is approximately normally distributed. That is the null hypothesis for this test. So basically we are going to make use of the SciPy start library, but we want to import the function Shapiro, and also from the SciPy start library we want to import the Anderson, right, function. So let's run this. Now I want to get that of the Shapiro-Wilk test. So I want to get the Shapiro-Wilk test of the sepal length for Setosa. The second one is going to give us that of versicolor, and the last one will give us that of the Shapiro-Wilk test of the sepal length for Virginica. Okay, so let's run this. So in here, the first result is that of the test statistic, and the last result is giving us that of the p-value. Now if we compare these p-values with an alpha level or a significance level of 1% or 5%, it means that we will fail to reject the null hypothesis. Okay, anytime you are using the p-value with an alpha value, we only reject the null hypothesis if the p p-value is less than an alpha level of significance. Now you can see that this p-value is greater than our alpha value of either 1% or 5%. So it means that we fail to reject the null hypothesis and conclude that the data of the sepal lines for setosa, for vesicular, and for vaginica are normally distributed. Okay, let's check that of the Anderson-Darling test. So we want to basically get the AD test of the sepal lines for setosa, for vesicular, and for vaginica. So let's run this. Okay, so in here we have the test statistic and the critical values as well as the significance level. This significance level are in percentage, so this one is 15%, 10%, 5%. The standard one is 5%, so I'm going to make use of the third values from my critical value. So if we want to make inference with regards to the test statistic and the critical value, then we are going to reject the null hypothesis if the test statistic is greater than the critical value. So the critical value at 5% is 0.73. Okay, this is the first one at 15% at an alpha value of 10%, and this third value is a critical value at an alpha value of 5%. So comparing this test statistic with that, we can see that this test statistic value is less than this critical value. So it means that we are going to fail to reject the null hypothesis and conclude that the data of the sepal length for setosa is approximately normally distributed, and therefore making use of the t-test is also required. Okay, so we can also check that of the vesicular. If you compare the test statistic with a critical value at an alpha value of 5%, right, we have this 0.736. We can see that the test statistic value is less than this, so we also fail to reject the null hypothesis and conclude that the sepal length for setosa is normally distributed, right, and therefore making use of the t-test is required. If you check that of the vesicular, for vaginica, you can see that the test statistic value is 0.565 compared to the critical value of 0.735. We fail to reject the null hypothesis and conclude that the data of the sepal length for vaginica is normally distributed, and therefore making use of the t-test is required. So now that we are certain that the normality assumption has been satisfied for this data, that is for setosa, for the data for vesicular and vaginica, we can then go ahead and make use of the parametric test, specifically the one sample t-test. Okay, so let's jump right in. So we want to make use of the parametric test because we have already satisfied this normality assumption. So for the one sample t-test, the null hypothesis is that the average value of the sepal length is 6, and the alternative is that the average value of the sepal length is different from 6. Okay, but before we make use of the t-test, we have to make use of the stat function from the SciPy library. So we should have imported this, but I've already imported the stat function. That's why I'm commenting on that. Okay, so we are going to make use of the stat function. I've already imported this, so I can skip that. Okay, so from the stat function, we're making use of the t-test, right? That's a one sample. My data is going to be that of the setosa, and I'm interested in the variable sepal length. The hypothesized mean is going to be 6, right? And the alternative is going to be two-sided, right? Because this is a two-tier test. If it was a one-tier test, then we are going to have less. Like if it's a left-tier test, it's going to be less. For a right-tier test, we are going to make use of greater. Okay, so let's run this for two-tier test. So we can see the p-value. I'm not so much interested in the test statistic value. So I want to compare this p-value with an alpha value of 1% or let's say 5%. It means that we are going to reject the null hypothesis, right, and conclude that the average value of the sepal length, right, for the setosa is different from 6 centimeters. Let's check that with the t-test of the sepal length for vesicular. Okay, so now my data is going to be gene2 underscore SL as the data for vesicular, and I'm interested in the variable sepal length. The hypothesized mean is going to be 6, and the alternative is two-sided. So let's run this. Alright, so if you compare this p-value, right, 0.384 with an alpha value of, let's say, 1% or 5%, it means that we will fail to reject the null hypothesis, right, and conclude that the average value of the sepal length for setosa is not different from 6 centimeters. Okay, now let's check that of the t-test of the sepal length for virginica. So let me run this. Okay, so if we look at the result of the p-value compared to an alpha value of 5% or 1%, it means that we are going to reject the null hypothesis and conclude that the average value of the sepal length for virginica is different from 6 centimeters. Okay, now assuming the assumptions that we tested for failed, then we are supposed to resort to the non-parametric test. Okay, so assuming the assumption that we tested for all failed, then we are going to make use of the non-parametric test, and specifically the test that we are going to make use of is the sign or the quantile test. Okay, so let's check that. So we need a library, right? We are going to make use of the stat models library, which is part of the SciPy library. So from the stat models stat library, we want to import the descripted stats as DS. Okay, you can change this abbreviation if you wish. So let's run this. Alright, so I want to make use of the sign test from the descripted stats library. My data is now G1, that's the data for setosa, and I'm interested in the variable sepal length. My hypothesized median is now 6. Okay, so for non-parametric test, the parameter of interest is always the median, not the mean. Okay, so let's run this to see the result. The first input here, or the first input here is basically a test statistic value, and the second input, right, in this result is the p-value. So I want to compare this p-value with an alpha value of 5% or 1%. It means that I'm going to reject the null hypothesis and conclude that the average value of the sepal length for setosa is different from 6, right? So this is basically confirming. It's also similar to the result that we had under the parametric test. Okay, let's check that of the sign test of the sepal length for vesicular. Okay, so let me run this now. My data is that of vesicular and the variable sepal length. The hypothesized median is now 6. Okay, let's run this. Okay, so if I compare this p-value with an alpha value of, let's say, 1% or 5%, it means that we fail to reject the null hypothesis. Okay, and we are going to conclude that the average value or the median value of the sepal length for vesicular is not different from 6 centimeters. Okay, and this is also similar to the result we had under the one sample t-test. Now let's check that of the sign test of the sepal length for virginica. So let me run this. Okay, so for virginica, we can see the p-value to be very small, right? If you compare this p-value with an alpha value of, let's say, 1% or 5%, then it means that we are going to reject the null hypothesis and conclude that the average or the median value of the sepal length for virginica is different from 6 centimeters. Okay, so I think we are good to go. So basically this is how we can analyze one sample data in Python using Jupyter Notebook. Please, if you find value in this video, don't forget to subscribe if you have not, and also turn on the notification bell to get more updates anytime I post on this channel. Thank you for watching.